Hey everybody, I'm Meredith. And I work here part-time. I'm mostly focused on the livestock, but I do work in one of our greenhouses, raising vegetable crops, and I've become really focused on soil and as well as water assessment with the microscope here on the farm, just to inform our management decisions. And everything we're going to be talking about today, I'm not taking any credit for it whatsoever. It's completely the methodology of Dr. Elaine Ingham, who came to visit a couple years ago, I guess, and did a class. And then one of her certified trainers then trained us, a few of us here on the farm. I'm going through her online classes now and hope to become certified really soon with her methodology. But that's kind of where this comes from. And if you're interested in more information about, once you leave today, that's the go-to place. Her business is called Environment Celebration, and it's her and Dr. Caroline Rollins partnering to educate people about the soil food web as well as proper composting and compostee methodology, and just contracting and consulting to build soil for people using beneficial biology, from what I know. We'll go through a quick PowerPoint. We'll talk about the different taxonomic groups in the soil that you might see under the scope. And I'll do a couple sample slides. We'll look at some samples together. Then we're gonna take a break until about 4, then we'll do breakout groups, and we'll have a good amount of time to do that. It'll be really free form, should be fun. At 6, Pat has prepared a meal, so we'll come together, we'll eat, and then hopefully once everybody gets their food, we'll get to share what we've seen, discuss about that. So I just made a quick list of all the things that we're hopefully gonna accomplish today. We're not doing any kind of quantitative process. I wanna be very clear about that. What we're doing is taking a look at the morphology or the physical characteristics of the organisms in your soil samples and getting a sense of what kind of life is there, how much diversity of life is there. We're gonna look at non-living clues that you're gonna be able to see through the scope for context about what's going on in the soil. You're gonna learn how to do that. We're gonna quickly be able to see the cumulative effects of soil management. So I don't know how each one of you manage your soil, but based on what I tell you, when you look through that scope, you might be able to say, I till too much, or something like that, So you'll have some really, really good visual cues that allow you to see how your management's affecting soil life. And then hopefully you'll use your new knowledge to think about ways to change. I can say I've been growing food on and off for 20 years, both as a commercial farmer, as a home gardener, working for a nonprofit. And I would say that nothing has solidified the way I manage soil and the management decisions that I make as much as looking at the soil through the microscope. It's been I would say as a veteran grower, you're constantly hearing different ways to manage soil or manage crops or use equipment. And you think, these are all just different options that I can use based on my preference or based on convenience. But taking a look at the soil has really changed my mind about some of that stuff. And I see certain management things as non-negotiable now in the way that I manage soil and the way that I manage crop plans. And so that's been really powerful for me. If you've never done this before, it's absolutely mind-blowing. I mean, just to see everything that's going on, it's going on all the time, but that realistically none of us ever get to look at. You know what I mean? It's really powerful to know how small we are, yet our actions have serious magnitude, right, on the earth for sometimes hundreds, 200 years. When you're thinking about the body of a fungus in the soil, if it has been there or never been there, or if it's dead or alive, the impacts of that are humongous for future generations. So this work is really exciting in that capacity. So let's talk about what you might see when you look through the scope. What we're looking at is different feeding groups, basically, different taxonomic groups of soil organisms. Bacteria are gonna be by far the most abundant organisms in the soil, and they, within their bodies, carry lots of nutrients for plants. So when they get eaten by things like Protozoa or nematodes, their nutrients in their bodies are being released for plants to uptake, right? So we need the bacteria, but we also need the things that eat the bacteria. What we're looking for is an intact food chain. In other words, so when we say the word soil food web, that's what we're talking about. We want a diverse, dynamic, living, dying, eating, pooping array of organisms in the soil. And that's going to inform the structure, it's going to inform the availability of nutrients, et cetera, et cetera. So going up the food chain a little bit, you're going to see protozoa, which are going to be, and I'll show you pictures in a little bit. So these are mostly things that eat bacteria. We're talking about flagellates, we're talking about ciliates, amoeba. Hopefully we're going to see some fungi. So these are really hard workers in the soil. They give the soil structure. They also, I mean, depending on the type of fungus, they can, you know, if we're talking about mycorrhizal fungus, which we'll talk about a little bit more detail, they're actually acting as a carrier system for plants, seeking water, seeking nutrients and other things. Really powerful stuff. Nematodes look like worms. If you've never done this work, you might think you have a parasite in your soil, but they're actually mostly good guys, unless we're talking about root-feeding nematodes, which actually can kill your plants, right? So there's several different types. We'll talk about how they're identified. You might see microarthropods are really not common in agricultural soils, but those are like, they're gonna look like big insects on your microscope slides. Kind of scares the dickens out of you when you see them because they're much, much bigger than the other organisms that you'll see. But they're kind of the choppers and the shredders in the system. You're also gonna see mineral components. So you'll see big, you know, humic acid, fuzzy looking, amber colored items. You're gonna see silica, you're gonna see clay particles, sand particles. You'll see organic matter, bits of cellulose, plant tissue, leaves, seed coats. And then you're gonna see a whole bunch of things that aren't any of your business and that we don't know how to explain and that probably matter, but we don't know why. So there will definitely be things where you're like, whoa, what's that? And no one in the room will have any idea. That's one of my favorite things about this, and I do a lot of food fermentation, and it's my opinion that our science can never really measure the synergy of what's going on the microscopic level, and so there's a great deal of that work that is none of our business. And all we have to do is provide the conditions, and nature does the rest. So let's look at some photos. This is a picture containing many things, but the most of what you see is a lot of bacteria. You can see the little round dots. You can see the rods. You can see slightly larger rods, slightly smaller rods. Lots of different shapes of bacteria. And I have a chart from Elaine coming up that will show you the different sizes. This is a pretty low dilution. So this is probably a 1 to 5 dilution. And we'll talk about that when we go to make our first sample. So it looks, you know, very, very heavy in bacteria. So there's some examples. Here's some different shapes of bacteria. So the cocci are gonna be the little dots. The diplococci is 2 dots together. This stuff doesn't necessarily matter for us today. The main take home is that you wanna see a lot of different types of morphology in the bacteria. The ones on the right side, the others, those are gonna be really characteristic of pathogen-causing bacterium, so you don't necessarily wanna be seeing those. But if you're seeing good diversity of morphology on the left-hand side, with a few exceptions, then that's a good thing, you know? Protozoa, the biggest thing on the screen is what I want you to focus on right now. That's a testate amoeba. And it actually looks like it might have very recently died in this photo because I don't see a lot of its internal plugs. So they'll just like take a plug out of the soil solution, and including bacteria, and then they'll sit there and digest the bacteria. It's very rare in a soil sample that you've shaken up to see an actual amoeba that is outside of a testate. So the testate is a protective little home for the amoeba. And a lot of times protozoa, you really need to learn how to, if you want to get into this work, you really need to learn how to identify the cysts of those protozoa because they go in and out of cysts. Cysts are protective environments for them, basically a membrane so that they're not gonna be toxic if they are a toxic organism, and they're also not gonna be killed or affected by what's going on in the environment. So say a livestock animal with a bunch of protozoa in its gut drinks some very, very cold water. It might shock those organisms in the gut, cause them to encyst. It doesn't mean they're dead. It just means they're gonna wait around until the environment is favorable again, and they're gonna come back out. But in the soil that we have scooped, chopped up, taken a clump of, put it in a test tube, put some water in it, and done a gentle shake on, you're mostly gonna be seeing, do you see these round blobs kind of on the left there and then on the right? So those are examples of cysts. And if they're super ornate and double-walled, in general, they're amoeba. And if they're smaller and single-walled, they're going to be flagella or they're going to be ciliates. So those are the three main groups of protozoa that you might see. The way to differentiate flagella from a ciliate is that, is everybody familiar with like the term flagella from, if you reach back to like 6th grade science, it's like this whip-like tail thing that comes off. I mean, think of a sperm. It has a flagella, right? So that's how it moves around. So if you see one that's not encysted, you might see that very faint whip-like structure, and that's how you'll know it's a flagellate. If you see a protozoa-looking structure with a bunch of tiny little hairs all over it, or tiny little hairs on the front or the back, that's going to be a ciliate. So the difference is cilia are little tiny hairs. You have them in your throat, to help food go down. Unless you're a smoker, you've burned them all off. And so the ciliates have the little hairs, the flagellates have the longer whip-like structure. So this is a really beautiful fungus that was in a pile of compost that we made over at our Grandview farm. What you're looking at is a fungal hypha, or the root. And they look like this. They generally have uniform diameter, and then they are branching. And if they're beneficial, you'll see little lines separating the cells. Those are called the septae. And so that's a good thing. If they have irregular septae, they're more likely to be oomycetes, or not so good fungus. But the wider the diameter, the darker in color, and the more uniform the septae, the more beneficial the fungus in general. So if you see something that looks tattered or frayed, but it's a strand, and you don't see septae and it doesn't have uniform cell walls, you're likely looking at a piece of organic matter or plant tissue. You're not looking at a fungus, right? So using those characteristics is how we identify. I think I put that one up so you could see the septa a little bit better. Do you see those little tiny segments between the cells? It's just the end of one cell and the beginning of another one. Fungi are so fascinating. I can't even get enough of them. So this is an example of an oomycete, or a not so great fungus. So see how those penicillin strands, they still have uniform cell walls for the most part, and you can still see septae, but they're clear, very thin. There are a mass of them. The little balls that you see are their little chlamydospores, or the ways that they're going to reproduce and grow. So if you saw this in a soil, you would be like, yikes, this is not a good thing, right? This is indicative of a really highly anaerobic situation. We won't be able to see this, but I wanted to talk to you about Mycorrhizae. All right, so there's a bunch of different species, basically, but it's a particular type of mutualistic fungus that grows in association with plant roots. And so basically, think of a little fungal spore. You'll see fungal spores today. They're usually dark and like oval shaped, or they could be segmented. But think of a little fungal spore, and a plant is releasing. So do we know what plant exudates? Do we know this term? So the plant photosynthesizes light from the sun, produces sugars. Almost 40% of that product, photosynthetic product, is actually pumped down into the soil in the form of a plant exudate. And it's basically liquid carbon. It's like sugar for the soil organisms, one of them being Mycorrhizae. So if a little fungal spore, Mycorrhizae spore, sprouts, it has about 48 hours. And this is a really interesting fact. It has about 48 hours to sniff out the exudate and make its way with its hyphae up to that plant root. And depending on the type of Mycorrhizae and the type of plant, it could colonize the outside of the root, which you'll see on the left there. That's an ectomycorrhizal association, which is mostly on conifers, so not really typical in agricultural situations, depending on, I don't know if anybody's a forester. And then on the right, this is more common for what you'll see in agricultural crops, is endomycorrhizal association. So that's where actually the fungus will grow inside and in association with the plant root. So the plant tells the fungus what it wants in terms of water, in terms of macronutrients, micronutrients, and the fungus sends out this extreme network of hyphae to go mining all that stuff from the soil, brings it back to the plant in exchange for continued plant exudates or sugars, which it then uses basically only to reinforce its cell wall. So, because everything else it's going and getting anyway on its own, right? So really powerful. You're gonna have much healthier plants and plant ecosystems if you have good mycorrhizal associations, because the plants themselves really aren't able to grow that extensive of root networks, especially in agricultural situations when we're going and plucking them out of the soil all the time. All right, so they just don't really have the opportunity to do that. They need the mycorrhizal associations. And there's a lot that we can talk about. If people have questions about like, oh, well, I buy mycorrhizal inoculant. Is it the right one? Or, whatever we can talk about. I don't know a lot of trade names, but I have done a fair amount of research about if you find out where they're getting it from, knowing whether or not it's going to be more effective or less effective. I just wanted to, you have to have like a UV situation going on with your microscope, or you have dyes in order to actually see the arbuscules, which are those little fluoresced bits inside of the root. You can actually dig a root and wash it in like a vinegar solution and then put it in India ink back and forth until it takes up the India ink, and you can look at it under a scope, and sometimes you can see the arbuscule on a plant. That's the one way to kind of see them that I know of, but I've never actually laid eyes on it. That would be very exciting. You might see what they call an extramatrical spore, which means it's outside the root, but if you wanted to see whether the root was colonized, you would have to have more capabilities than what we have right here. Here's some nematodes. As you can see, they're much, much bigger than the other organisms we've looked at. So, and in general, they move very, very fast. So they're hard to miss if you have one or two on a slide. For the most part, they're good guys. They eat different things. There's whole groups of them that eat bacteria, which is great. We need something to keep all that bacteria in check. Then we have ones that eat fungi, and then we have ones that eat each other, and then we have some that spear our plant roots and kill them. And so if you want to get rid of the ones that spear the plant roots, you have to have the ones that eat other nematodes, right? See what I'm saying? So we need a good balance. In general, it's a higher taxonomic group. So in highly disturbed soils, you're not going to find hardly any nematodes at all, and definitely not fungal feeders. Or I mean, think about it. If there's no fungi in your soil, you're not going to have fungal feeding nematodes, right? Depending on what you're looking at. If anybody brought worm castings, I almost always see nematodes in worm castings when I look at them. But you'll probably see some bacterial feeders in there. And I have a few more pictures, I think. The way we identify nematodes is by looking at their mouth parts. And this is probably something that we won't get into today. If we see a nematode, we will try to identify what kind it is, but don't worry about going into big detail about memorizing all the mouth parts today, because I still have to refer back to this on a regular basis. But the way it's differentiated is things like the medial bulbs, see the bulb there after the mouth, and then the terminal bulb after that. So you'll almost only see a medial bulb and a terminal bulb on a bacterial feeder. If you see a nematode with fancy lips, like the ones up there, some of them are very fancy indeed. It's almost always a bacterial feeder. Fungal feeders have a lesser defined bulb. Sometimes they won't have one. And then they have a very tiny little spear. See that spear up near the mouth where they inject it into the fungal hyphae. Root feeders have a very strong spear, and it has a little knob at the end of it that's pretty discernible if you're looking at a root feeder under the microscope, because they have to have a lot of strength for shooting into the root and killing it. Predatory nematodes have that weird tooth thing right there near the mouth and all that skeletal looking stuff. And then omnivores, so those are really interesting. They normally feed on fungus, but they'll feed on other things too if there's no fungus there. And some of them will actually switch over and start eating roots, which is not awesome. So the point there is if you have omnivores, they're not bad guys unless you're doing something wrong and you don't have fungus for them to eat, right? So, yeah.
Are the fungal nematodes considered bad since we're trying to enhance the fungal communities in our soils?
If you're not doing anything to foster fungal populations in your soil, those two groups are gonna, it seems like, keep each other in check, right? If you run out of fungus, you're gonna run out of the fungal feeders. But you do want balance on all of your taxonomic groups. So if you have fungus, you do wanna have some fungal feeding. Nematodes, there's also fungi that traps nematodes, which is really cool. I didn't upload any photographs on it, but it's one of the most amazing things I've ever seen in my life. And the first time I heard about it, I was just like, wow, people don't matter at all. So it's like this fungus, there's a couple different kinds, but one is like, it creates these little lassos, and that the nematodes like even put a piece of their body like near the lasso, it'll constrict and trap them. And then it just like grows into them and kills them. And it will kill any of these. It doesn't care. But it's one of the ways of keeping some of the other nematodes in check, you know? So nature sort of balances. Yeah.
You say that, I mean, I've always been taught, in general, fungal dominant soil is what you're kind of looking for.
Yeah, so, I mean, fungal dominant soil is typical of permanent ecosystems like forests. But the more we can foster mature soils in agricultural situations, the more carbon we're sequestering and the more nutrients are going to be available to plants. And so one of the biggest impacts on me of this work is understanding that even in alternative agriculture, there are things we've borrowed from convention that are not ideal. Continual soil disturbance keeps soil in an early successive state. Where you will not find fungal communities, you will not find higher taxonomic groups, and you will find heavily bacterial situations. And I was going to talk about this later, but we can talk about it now. But what that means is, think about a roadside that's been blasted. What do early successive plant species look like on that soil? They look like weeds and invasive groups, right? If you are constantly disturbing your soil, you're going to get early successive organisms there. Mostly bacteria. Overgrowth of bacteria equals weeds, equals crusting of the soil, equals all these things that we consider unfavorable in organic agriculture. So yeah, that really hopefully turns on a lot of lights for you about management, which we can talk about as we go. Here's an example of a bacterial feeder. I always get excited finding bacterial feeders in agricultural soils because I just want to find things to keep bacteria in check. I want to see higher taxonomic groups. I get excited when I see fungi. I get excited when I see bacterial feeding nematodes. I'm working on completely undisturbed compost right now, which may show me some higher fungal feeding nematodes and some predatory nematodes and things like that. The only time I've ever seen a fungal trapping nematode or an predatory nematode is in some like ancient leaf mold that Pat brought me from his house in Silo that no one had touched for many, many years. And it's like probably the most mature thing I've ever looked at in terms of a soil. But this is a bacterial feeder. I just wanted to show you, like you just try to get as close as you can to the mouth part. It's very hard to do. They're moving very fast. You have to change the focus. Because even if you have smashed your soil droplet of soil sample under a cover slip, it's still 3 micrometers deep. And microscopic organisms are moving around within that depth. And so you're going to have to be focusing in and out through the depth.
Have you noticed different soil structures or types to harbor different nematodes?
Different soil types to harbor different nematodes? Definitely. You mean like silt versus sand versus clay? Well, I haven't tested a lot of soils where I knew the order, I guess. But from what I know about what causes these organisms to flourish, soils with higher mineral content like clay soils are going to have more life in them than sandy soils, for example, that leach nutrients and leach water. And a super clayey soil that's really compacted is going to be anaerobic, which is not going to foster these kinds of lives. So the mineral content of the soil definitely affects it, and the physical characteristics of the soil are going to affect it. Most of these organisms will cease to thrive at lower than 6 parts per million oxygen. So in general, as you move towards anaerobic conditions, you're going to see less favorable organisms. And the answer is going to be aerate, right? That's like really simple way to get the life back into the soil, first and foremost. All right, so here's an example of some of the like non-taxonomic organism things you're going to see. Mineral components like the clear, really crystalline, refractive things are going to be examples of like sand. Sometimes if they're very geometrically exciting, they might be minerals like from fertilizer or something that are floating around in the soil. If you're seeing a lot of that, it means that you have a salt problem and that you're feeding too many simple amendments all the time with all these loose minerals in them. And what you need to do is just chill out, let the life in the soil unlock everything and send it out to the plants. So these kinds of things are what are fascinating to be able to see. The big brown fuzzy things are aggregates or organic acids. So humic acid is that color. That's a great thing to see. If you're seeing aggregates in your soil, it means that you have decent aeration, right? So one of the things I didn't mention about the ciliates in that group of protozoa I was talking about, the flagellates, the ciliates, and the amoeba, the ciliates are kind of like here and there, no problem, but if you see huge amounts of them, it means you're heading anaerobic. So other clues in a really anaerobic situation, if you're like, I see a bunch of little critters moving around really fast, I wonder if those are ciliates, and you're not sure because you can't see their little cilia or you're just starting out, well, do you also not see a bunch of aggregation, right? Do you also see a bunch of lactobacilli, which is an anaerobic bacteria that's really long and skinny? So all these context clues, the living and the non-living, come together to give you a picture and help you understand what's going on in your soil. So if we're not seeing aggregation like that, probably need to get a little bit more air and a little more structure and work on the structure of the soil. And I think that that's one of the things that our compost was suffering from when I first started looking at it, is that we were overworking it. We were turning it far too much. And so we were not only just obliterating any fungal hyphae that were there, but we were also destroying structure and creating more of it wasn't anaerobic, but it was leaning that way, and it was just way overgrown with bacteria. And so it looked really good. It was crumbly, it was dark, it had all those visual cues that you associate with good soil, but when push came to shove under the scope, there really wasn't much to it at all.
Remind me of how humic aggregates are formed again.
Well, it's glue called glomalin, basically, that comes from bacteria what's that?
Doesn't that come from microorganisms?
It does. It comes from fungus, but I think it also mostly comes from, all bacteria create glue. Some of it's really acid and some of it's not. So anaerobic bacteria are going to create a glue that's not so awesome, it's an acidic. But the good bacteria are going to create a nice alkaline, good, gluey substance that pulls together carbon, stable carbon, into these clumps. And then all the little guys like to live on and around those clumps. And so if you just want to look at the soil and see exciting things, zone in on your aggregates, because you'll see around the edges, I've seen ciliates scurrying all around an aggregate and just vacuuming all the bacteria off the top of it. Or I looked at some compost tea from the season the other day, and I saw these little baby bacterial nematodes. They were so cute, and they were just I guess they were the larval form, but they were just scooting all around, going really fast, but I was able to just follow one of them, and he was just going all around that aggregate. It's amazing the depth that I was witnessing in this tiny little soil sample. He was just having himself a feast,
Fisherman on coral reef.
Yeah, exactly. Here's just a few examples of the types of plant tissue and different stuff that you'll see. It'll be whoa, I found something no one's ever discovered. And it's no, it's actually cellulose. So the curlicue thing is almost always going to be cellulose from wood or paper. The top right is actually a seed coat that has fungal hyphae growing over it, and it's being decomposed. And then the thing below that looks like the trachea of something is likely just plant tissue of some kind. And you'll see all kinds of things like this, where, I found this one thing one time. It was round, and it had these two little round ears kind of coming off of it. It was about this big, and I sent it to my mentor, and she said, "It's the famous Mickey Mouse pollen of the blah, blah, blah conure." I was oh my, I have no idea what you're talking about. So we can go back to that slide later. We should probably just go to the scope now.